Okay, this is a hopefully brief screencast that will get across to you some of the ideas we were trying to get across to you yesterday in class, and I think we serve to confuse you more than clarify things for you. So I want to go through and try and rectify that today. So we were talking about adaptive landscapes, and in particular, Dr. Reynolds was trying to get across to you this idea about the relationship between the trait of survival and the trait of reproductive output, and get across to you the idea that there are constraints involved in the evolution of organisms, and was using this guppy example. Now, it turns out that when you think of survival and reproductive output as traits, in guppies, those are also correlated with size. But let's not think about size for the moment. Let's just think about the two cases of one trait being survival and the other trait being reproductive output. So you can have low survival or high survival, and you'll recall that big guppies had lower survival than little guppies, which had higher survival. And you have reproductive output, which you can have low and high. And in guppies, reproductive output is correlated with size, and so big guppies have high reproductive output, and little guppies have low reproductive output. So out in nature, if you're small, you have high survival, but you have low reproductive output, which puts you down here. And in nature, big guppies have high reproductive output, but low survival, which puts you up here. And so we can basically draw a line of highest fitness that basically shows that along this line is the region of highest fitness. And so you can have intermediate survival coupled with intermediate reproductive output, but in order to get the highest reproductive output, you have to sacrifice some level of survival, and to achieve the highest survival, you have to be small enough that your reproductive output isn't very good. And so this represents a constraint whenever you have this negative correlation. And as you move away from this sort of situation, you experience lower levels of fitness. And so this is what an adaptive landscape might look like, where you have a ridge of high fitness surrounded by areas of low fitness. These are low fitness because you don't really achieve anything, not surviving well and not reproducing very well. And you don't achieve this because it's impossible to have everything. And so that's where this idea of constraint comes in. So you have an area of high fitness and areas of low fitness. And this idea of constraint is kind of universal in evolutionary biology. We see it in size number trade-offs, where the more offspring the bigger your offspring are, the fewer offspring you have. And that just represents another kind of constraint in biology. And so when we think about constraint colloquially, we can think about it as, for every advantage there is a disadvantage. You can't have your cake and eat it too. You could be Superman, you have super strength, you can fly, you can have laser vision, you can see through walls, you can date Lois Lane, but you're susceptible to kryptonite. Or you can think of areas of constraint in your own life. I can study for this exam, or I can go see a movie. I can't do both of those effectively at once. In biology, we think about evolutionary constraints, and these constraints can be morphological. By specializing one part of my morphology to do something well, it means that I can't do other things as well. There are physiological constraints, there are energetic constraints. The one that we just looked at was an energetic constraint. And then there are genetic constraints, because things are genetically correlated with one another. And so this brings us to an example from Little Butch Brody's dissertation work, where he was looking at this species, Thamnophis organoides. And Thamnophis organoides is a species that occurs in the Pacific Northwest. It's a species that doesn't feed on newts. It feeds on worms and slugs and things like that. And the interesting thing for him was the fact that in some cases Thamnophis organoides looks like this with a very striped pattern, and in other cases it looks like this with a very checkered pattern. And his question was, why would these two color patterns be occurring, and they even occur in the same pasture, in the same population, in the places where he was studying these in the Pacific Northwest. And so one of the things he was interested in is things like performance, but his main question was, how can this polymorphism, this color pattern polymorphism, be maintained? Well, when he and his dad raced these snakes down racetracks, and here you can see Edmund Brody Jr. racing a snake down a racetrack lined with AstroTurf, he would notice that some snakes would just run straight down the track without any problems, and then other snakes would tend to turn around and reverse themselves. And he marked down the behaviors as well as marking down the speeds, and he also created a way of quantifying color pattern so that he could place a number on a snake that would tell you how relatively stripy it was and how relatively checkered it was. And then he took babies of these snakes, raced them down racetracks, recorded their color pattern, and then released them to the field, and then went back at the end of the first season to look and see who survived and who didn't. And what he found was that here's the reversals axis here, here's the stripe axis here. Stripe goes from a negative value, which is very checkered, to a high value, which is very stripy. And reversals goes from a negative value, which means that you tend to run in a straight line, to a high value, which means you tend to turn around on yourself quite a lot. And what he found was that in terms of survival, and so fitness here is basically survival, he found that checkered snakes and striped snakes can both survive, but checkered snakes survive best only if they also tend to reverse themselves, and striped snakes tend to survive only if they tend to run in a straight line. Checkered snakes that run in a straight line and striped snakes that tend to reverse themselves a lot end up not surviving well, and actually striped snakes that reverse themselves a lot have the lowest survival altogether. And so what this means is that the color pattern and the behavior are negatively correlated with one another, such that you can either be checkered and have a particular behavior, or you can be striped and have the other behavior, but you can't be checkered and have both kinds of behaviors. And so, and down here you can see that the fitness is such that there's directional selection on both stripe and reversals, and we'll talk about this in a few minutes when we start to talk about quantitative genetics of it. But another way of thinking about this is, imagine that you are a population of snakes, and in that population, you have a population of snakes, and in that population of snakes, you can have all possibilities of color pattern and behavior. But you set this population out into the world, and what you have is this adaptive landscape that exists out there in the world, where your highest fitness is when you're very striped but don't reverse yourself very well, or you are checkered and you tend to reverse yourselves. Well, imagine that there is a fitness line here of 0.5 fitness, and to the upper left and lower right of this is lower fitness, and to the upper right and lower left is higher fitness. What happens over time is everybody in the low fitness range tends to go bye-bye because they tend to get caught and eaten by predators. And so you lose those individuals from the population, and in reality, in the end, you'll lose these individuals from the population as well. But I'm leaving them there for a moment because we can get rid of the adaptive landscape and we can get rid of these fitness lines, and what we find is this negative correlation where you have checkered snakes that reverse themselves, and you have striped snakes that don't reverse themselves, and you have a range of individuals in the middle along this kind of ridge of high fitness with low fitness to either side of that. And this leads to a phenotypic correlation that you could observe by just going out, collecting some snakes, and running them down a racetrack and scoring their color pattern. And so this is what the three-dimensional fitness landscape looks like. And you can see this ridge of high fitness right here, where the best thing to be is a striped snake that runs in a straight line. The second best thing to be is a checkered snake that reverses itself a lot, and the other combinations have relatively low fitness. If you collapse this grid down onto this flat plane, it ends up looking like this fitness surface. So we modeled before the evolutionary response to selection by basically saying the evolutionary response to selection is due to heritability multiplied times the strength of directional selection in this case. And this is good because you can actually now put numbers on these and see how fast you expect a population to be evolving. But this only works for a single trait. And in this case, we're actually dealing with two traits. We're dealing with stripedness, and we're dealing with this behavioral trait reversals. And so we need to expand this model out to include the evolution of two traits. And so the way we do this is basically by quantifying the degree to which each of these traits is heritable, and we also quantify how these two traits may or may not be genetically correlated with one another. And so we come up with a new model that is basically just an expansion of the old model. So instead of having one trait that is responding to selection, we have two traits that are responding to selection. This is the additive genetic variance, which Dr. Reynolds talked to you about in class. This is essentially the heritability of trait 1. This is the additive genetic variance of trait 2, which is essentially the heritability of trait 2. This is the degree to which the two traits are correlated with one another. This is known as the additive genetic covariance. And you can measure this in a similar way that you measure heritability by just regressing the parental trait 1 against offspring trait 2. And so these will be the same number and will have the same sign. Beta 1 and 2 is directional selection acting on trait 1 and directional selection acting on trait 2. And so these are basically taking the place of S in the previous equation. And so the way this works, this is matrix algebra. G11 times beta 1 plus G12 times beta 2 quantifies what's happening to trait 1. G21 times beta 1 plus G22 times beta 2 quantifies what's happening with trait 2. Now in this case, selection on trait 1 can affect trait 2 through the genetic correlation of trait 1 and trait 2 in the same way that selection on trait 2 can affect trait 1 through the genetic correlation of trait 1 with trait 2. And so this leads to some odd evolutionary dynamics. But let's just think about this in the simplest case where we have, I've color-coded everything so you can follow the math a little better. But basically we have directional selection, and if you look back at the fitness landscape, you'll see that these are the numbers directly from Brody's paper. So this is directional selection on reversals. This is directional selection on stripedness. And so that's beta R and beta S. You multiply that times the genetic variance covariance matrix, and in this case this is actually the additive genetic variance for reversals and the additive genetic variance for stripedness. And in this case, I have just arbitrarily set the genetic covariances at 0, so the only thing acting on these two populations is the effect of direct selection. And this equals this thing, so we basically take GRR times beta R, which is 0.245 times 0.08, and GRS times beta S, which is 0 times 0.102. Those two things add together to basically quantify the direction that reversals will be going. And you do the same thing with the latter part of the equation, which is GSR times beta R and GSS times beta S, which appears down here. This thing reduces down to this, and then that basically equals, we should expect reversals to move a little bit, we should expect stripedness to move more, because stripedness has both higher selection and a higher degree of heritability than stripedness, than reversals, which both has a lower selection acting on it, a weaker strength of selection acting on it, and a lower heritability or additive genetic variance. So if you want to think about this, we could actually put this on a graph where we have reversals from few to many and stripedness from less to more. And when we look at the effect of selection acting on reversals, we see that reversals will increase a little bit and stripedness will increase way more. And we end up moving to a new spot because of the combined effects of reversals and stripedness. So we're now at this combination of traits, and we are now moved to a different combination of traits. But let's look at the situation that Brody actually found, which was that there was this level of selection, but the genetic variance covariance matrix was such that these two traits are negatively genetically correlated with one another, which means that as you get stripier and stripier, the genes for stripe are correlated with the genes for reversals, and as you get stripier and stripier, you tend to reverse yourself more and more. So, is that right? No. No, as you get stripier and stripier, you tend to reverse yourself less and less. That's how it works. Sorry about that. So if you work the math, you still are taking this times that and this times that, but now this is not zero, it's negative. And so this is the effect of direct selection acting on the additive genetic variance of reversals. This is the effect of selection on stripe acting on reversals. And this leads to a very different result, because now we have direct selection acting on reversals and indirect selection acting on reversals. One is a positive, one is a negative, and the negative is much bigger because the negative genetic covariance is relatively strong and selection on stripe is relatively strong. And so what we get in the end is we find that selection on stripe is driving the evolutionary change in reversals in exactly the opposite direction of what you would expect selection to be taking you. And so another way of thinking about this is to just put it back on this same plot that we had before, where we have direct selection acting on reversals, moving reversals in one direction. We have direct selection acting on stripe, moving us in another direction, which takes us to there. But we also have to consider correlated selection acting on other traits that are correlated with the traits that we're looking at. And so we find that the effect of directional selection acting on stripe affecting reversals takes reversals back the other direction, and direct selection acting on reversals ends up reducing stripe a little bit. And so we actually end up getting over here such that the overall response to selection is that we have an increase in stripedness and a decrease in reversals. And over time, if this happens enough, you get stripier snakes becoming less and less reversing, and you get more checkered snakes doing reversals more often because of the structure of these genetic correlations. So multivariate evolution is interesting because negative genetic correlations can cancel out or slow down evolution. Positive genetic correlations can speed up evolution. And in general, the direction or rate of evolution is determined by the shape of the adaptive landscape that an organism is living in, that a population is interacting in, and the underlying genetic architecture of the traits that are the object of selection. If those traits are uncorrelated, then evolution is relatively free to move the population to wherever selection is taking it. But if genes are correlated, oftentimes by sitting next to one another on the same chromosome, or by being linked through things like physiological pathways, molecular pathways. That underlying genetic architecture might be constrained because now those traits are correlated with one another. So we talked about these constraints earlier, and I'll just provide you some examples. One of the morphological examples, and this is from some human data, is the Carn-Penrose data from 1951. This is the probability of survival versus the birth weight of babies. And preemies, premature babies, don't survive very well, mainly because they're premature. They haven't undergone their full development and they're not ready to be born yet. But then there's also this morphological constraint up here at the top, where if you're too big, you would survive just fine if only you could fit through the birth canal of your mother. And so this is once again an example of balancing or stabilizing selection on birth weight in humans because of a developmental constraint on this side and a morphological constraint on this side. You have energetic sorts of constraints. This is an example that I had earlier in the presentation where you have a certain amount of energy that you can put into reproduction. You can either divide that up into large offspring, but because they're large, you can have relatively few of them, or you can have small offspring, and as a result, you can have a lot of them. But what you can't have is you can't have a lot of really big offspring because you just have a finite amount of energy to put into reproduction. And then we've talked about the genetic constraints, like what is going on in the garter snake Thamnophis organoides. And hopefully this clarifies some of what we probably confused you about the other day in class. So I hope this helps, and let me know if it doesn't.